All right, cool. So let's now go ahead and begin part 6.4, which is going to be molecular signaling within the neurons, as well as an overview of a handful of psychiatric conditions that are implicated in these types of molecular signals and these different types of pathways. So we're going to talk about that towards the very end. Again, I restructured this presentation and I had to move some things back around. So psychiatry is going to be towards the very end of this. So hopefully we'll be able to get to it today. I think we will. So we should probably be able to finish this by the end of the day. So these are the things that we're going to be covering in this section. We're going to talk about different types of chemical signals. We're going to talk about, for example, signal amplification as well as second messengers. We're going to briefly discuss the difference between impermeate versus permeant molecules, meaning that they either bind to the surface receptor or they go through the phospholipid bilayer, or cell-associated molecules, which means that you have two cells basically coming into contact, and you have a ligand on one cell and a receptor on the other cell. We're going to talk about different types of receptors. We're going to also go into more detail on what G-coupled receptors actually do. And then we're going to discuss protein kinases and phosphatases. Briefly discuss dendritic spines and what those actually do in terms of either being a place where you warehouse different components within the cell, as well as to slow down certain types of signals to reduce excitatory postsynaptic potentials. Then we're going to briefly discuss transcription regulation and transcription factors, because a lot of these second messengers, like the ultimate goal, is to create transcription factors so you can enact different types of transcription and then translation of proteins. And then we're going to go into psychiatric conditions at the end, focusing on schizophrenia, some of your mood disorders like bipolar depression, and regular MDD, which is major depressive disorder, as well as some anxiety disorders as well. And I put a little image here that I made using Mid-Journey of one of my favorite psychotherapists from back in the day, Dr. Carl Jung, really a huge pioneer within the field of psychotherapy. He was a medical doctor. Him and his contemporary Sigmund Freud basically created what we know today as psychology, like modern psychology. So Carl Jung, really interesting dude. I like him better. I like Freud a lot, but I like Jung a lot better. He was very esoteric in his thinking and approach on psychiatric conditions. So really fascinating character. Yeah. Unfortunately, we're not going to go too much into those types of psychotherapeutic approaches and whatnot. We're just going to cover the broad ideas of major psychiatric disorders. So how do cells communicate with each other? We've already discussed some of these, right? So we talked about synaptic communication, right? That involves neurotransmitter release into a synapse that can bind to a postsynaptic receptor to allow for the postsynaptic cell to actually do something, right? It can either be excitatory or inhibitory, as we've already learned. Paracrine communication, we're not going to go into that so much in this class. It's not as relevant, but that's where you have chemical signals that are within a tissue communicating with each other, right? But it has to be within the same type of tissue. And the word para, if you see that as a prefix, para just means adjacent to, right? Think about a paralegal, for example. A paralegal works with an attorney, right? Does all kind of the paperwork and all that, the regular day-to-day business, but the attorney is the one that does all the major type of work, right? So paracrine means adjacent communication. Then you have endocrine, which is going to involve hormones. Endocrine, generally speaking, is going to involve communication from one endocrine gland to a target organ far away, and it's going to be involving the bloodstream. So you have a hormone that gets released into the bloodstream. The hormone travels around until it finally comes in contact with a receptor. And once it contacts a receptor, then it's going to allow for different types of cellular changes. So what could those changes be? It could be, for example, a synthesis of proteins, right? Think about your sex hormones, like testosterone, estrogen. The goal of those hormones are to synthesize different types of proteins, right? And they're going to be produced in different areas of the body so they can enact changes in different parts of the body. And then you have intracellular signal transduction, and that's going to be signals that are going to be transmitted within the cell. And so that's kind of what we're going to be focusing on today. So here's your synaptic transmission. So you got your neurotransmitters being released by the presynaptic cell, enacting an effect on the postsynaptic cell by binding to receptor sites. Then you have your paracrine signaling. It's going to be within a tissue. So it's cells that are adjacent to one another within a tissue. And then you have your endocrine signaling, which again, you have an endocrine gland producing a hormone which gets into the bloodstream and affects a target organ far away in the body. Now, with chemical signaling, you have to have synthesis of the proteins and the neurotransmitters, for example, that can be released. You have to have specific receptors that will be able to bind to those ligands. And then you have effector molecules within the cell. This is kind of what we're going to be talking about today, is these effector molecules, and then the actual response, which oftentimes involves DNA synthesis, RNA synthesis, so transcription, and then ultimately translation of different types of proteins. So here is an example of signaling pathway. So this is signal amplification. When you have signal amplification, all you need is one single ligand to bind to a receptor to have a massive effect. I'm talking like 1 million to 1 billion fold increase of proteins just from one single ligand binding to a receptor. So it can go up to 10 to 1,000 fold, and then phosphorylation of the transcription factors allows for a massive increase of that signal. It's basically a domino effect. Absolutely. That's a great way to think about it.
How does it amplify?
This is how it does it. So you have the one ligand, it binds to a receptor, and then it's going to allow for G proteins to get activated. And then the G proteins, when they activate adenylyl cyclase, that's not going to involve amplification. But once adenylyl cyclase is created, then that's going to amplify cyclic AMP. Cyclic AMP is going to affect protein kinases. And then what do protein kinases do? They basically phosphorylate proteins. So you have phosphatases, which remove an inorganic phosphate from a protein so it can allow for deactivation. But if you phosphorylate a protein, that activates certain proteins. And so when you have activation of certain proteins, you can have a massive intracellular effect, which can also allow for things like gene transcription to translation of proteins. And so G-coupled proteins are going to be one of the major components of this entire equation. And cyclic AMP is also going to be a major component to this entire equation. So let's go ahead and watch a video that does a really good job at describing how G proteins work.
G protein-coupled receptors, GPCRs, have central roles in many physiological functions and diseases. This diverse superfamily includes over 800 members, all sharing a common configuration, passing through the membrane 7 times. GPCRs are present on all cells in the body. They pass signals from a variety of extracellular messengers to modulate intracellular pathways via the activation of G proteins and other signaling molecules. GPCRs mediate an incredible array of essential actions in the body, including smooth muscle relaxation and contraction, neurotransmission, immune regulation, and overall metabolism, and thus have enormous potential as therapeutic targets for multiple diseases. GPCRs are the largest and single most important family of drug targets in the human body. 25 to 30% of current drugs target GPCRs, including many of the best-selling drugs. Despite this, drug development has been encumbered by a lack of structural and mechanistic information about GPCRs and how compounds interact with them. The predominant problem is that GPCRs are too unstable to easily purify, and they lose their highly organized structure and activity once removed from the cell membrane, precluding them from use in structure-based drug design. To overcome this problem, Heptares developed a structure-based drug discovery (SBDD) platform to transform GPCR drug discovery.
Okay, so now that's turning into a commercial. So let's now watch another video that is going to describe what cyclic AMP also does in terms of intracellular communication pathway.
First, the signaling molecule adrenaline will migrate to the cell surface and bind to the G protein coupled receptor. The G protein coupled receptor will undergo a conformational change, which encourages the alpha subunit of the G protein to release its bound GDP and bind new GTP. Binding of new GTP will activate the alpha subunit. The alpha subunit will then activate the membrane-bound enzyme adenylyl cyclase. Adenylyl cyclase will turn ATP in the cytosol into cyclic AMP, or CAMP. CAMP will then bind to the inhibitor proteins on protein kinase A and release them. This activates protein kinase A and a phosphorylation cascade. Protein kinase A activates phosphorylase kinase using ATP. Phosphorylase kinase then activates glycogen phosphorylase in the same manner. Glycogen phosphorylase will lead to the breakdown of glycogen to be used as energy by the skeletal muscles during the fight or flight response.
So one type of example of what G protein coupled receptors do and then how they activate a downstream cascade. So that's an example in skeletal muscle, but you see that in all sorts of cells within the body. They all have kind of a similar effect in terms of intracellular communication, but they often use different types of protein kinases. So we're going to talk about some of those different types of protein kinases in a little bit, but they all do something different, right? In terms of neuronal communication, it's going to sometimes involve creation of proteins to allow the cell to develop and grow, or like even to do other types of things that involve transcription and translation of proteins. So very cool stuff. Now, here are a couple different types of molecules. So you have cell impermeant molecules, meaning that they do not permeate through the surface of the cell. So in other words, they need to bind to a receptor protein that's going to be found on the surface of the cell. So synapse, that's going to be a great example of that, right? A neurotransmitter binding to a receptor site. Versus a cell permeate molecule that does cross directly through the actual surface of the cell, right? It goes through the phospholipid bilayer. And these are often going to be things that are lipophilic, right? So if you have a lipophilic type of compound, that means it's going to be able to go right through the phospholipid bilayer, right? Whereas if you had like a hydrophobic molecule, it's not going to cross, right? A hydrophobic molecule is going to have to bind to a surface receptor. So sex hormones, steroid hormones, estrogens, things like that, those are going to be cell permeate molecules. They can permeate right through the phospholipid bilayer. And then the last one, which we're not going to talk about too much here today, but cell-associated molecules. That's where you have like two cells. One has the key, the other one has the lock. They need to come in direct contact with each other to be able to enact their effect. So integrins would be an example of something like that. Yeah.
I was thinking, you were saying something about hydrophobic molecules not being able to go in, even hydrophilic, because it can't really go in because of its polarity.
No, if it's oh, sorry. Yeah, I take that back. You're right. Yeah, so hydrophilic. Yeah, so if it's water soluble, hydrophilic. If it's hydrophilic, it wouldn't be able to cross. Thank you for pointing that out. Yeah, so apologies, I misspoke. So lipophilic crosses, hydrophilic does not cross. You're absolutely right about that.
I'm an advanced biochem, so
yeah, good. You're smarter than I am. That's perfect. No cellular response can happen without a receptor. And these are all the different types of receptors that you might find in a cell. You have those ion channel linked receptors. We talked about those already when we were discussing membrane potentials, right? So when you have for example, like GABA, when you have GABA receptors being bound, it's going to open up, for example, chloride ions, and then it allows for hyperpolarization of the cell. So they are going to be allowing for ions to move in and out of the cell. Then you have your enzyme-linked receptors, so you're going to have enzymes that allow for a chemical reaction to take place. So it's going to be involved in a cascade of intracellular communication. G protein coupled receptors, right, those are going to involve second messengers. And the second messengers are going to allow for, again, another type of intracellular cascade to take place. Cyclic AMP, adenylyl cyclase, all that stuff. And then you have your intracellular receptors, and those ligands have to be able to cross the phospholipid bilayer. So they have to be lipophilic. Otherwise, they're not going to be able to get through into this, to the inside of the cell. So here are your G proteins. So you have a couple different types. I'm not going to stress this very much. This is more like for your information. If you understand the basic premise of a G protein coupled receptor, you should be good. But the heterodymic G protein complexes, this could be associated with an alpha subunit being activated, and then after that gets activated, then you have a cascade involving cyclic AMP and then protein kinase coming into play. Whereas with a monomeric G protein, it's going to involve the RAS activation, which also activates MAPK. So these are two different types of G proteins, but like I said, I'm not going to make you guys responsible for that. So here is the cascade again. When you have your G proteins being activated, they are going to involve, again, second messengers. And there's a bunch of different types of second messengers, which I'm going to show you in a chart. But you have adenylate cyclase being produced. So once you have the receptor being activated, you get the G proteins, which get activated. You have adenylate cyclase. And then in the presence of ATP, it has to have ATP present so you can create cyclic AMP. Without ATP, you're not going to be able to have that effect. And then with cyclic AMP, that's when you get the expression of your protein kinases. Again, protein kinases phosphorylate proteins. So they take an inorganic phosphate and then they attach it to a protein, and that's what activates those proteins. These G proteins can also be associated with ion channels. So with muscarinic activation, so when you have muscarinic receptors being activated, those are going to open up potassium channels. So it doesn't necessarily have to involve cyclic AMP and protein kinases. It could also just open up an ion channel. So here are a couple of different examples of G protein receptors. So norepinephrine binding to adrenergic receptors. Then you have protein phosphorylation downstream. Protein phosphorylation is going to be the ultimate target. And all the intermediaries involve adenylate cyclase, cyclic AMP, protein kinase, and then the protein kinase is what allows for phosphorylation of proteins at the end. Glutamate can involve a couple different pathways. It can involve IP3 or it can involve protein kinase C, calcium, right? So different types of cascades could be in effect. Dopamine is going to have a reverse effect. It's going to turn off the cascade. So it's going to have an inhibitory effect on this cascade. So dopamine will reduce adenylate cyclase, will inhibit cyclic AMP, inhibit protein kinase, and then will decrease phosphorylation downstream. So they all have different types of effects. Yes?
You were saying a second ago the ATP produces a cyclic AMP.
Yes.
Cyclic AMP causes protein kinases
Yes.
to be made.
Yes.
And then protein kinases take an inorganic protein
Yes.
and attach it to
Inorganic phosphate.
Okay.
Inorganic phosphate. So it takes an inorganic phosphate and it attaches that to a protein, and that's where you get activation of a protein.
Okay. Thank you.
And then those proteins could act as like transcription factors, which we'll talk about in a little bit. And those transcription factors can go into the nucleus, allow for transcription of RNA, and then ultimately translation into proteins. And so here are a list of your second messengers. So calcium is going to be a very, very common second messenger in a lot of cells, and it does a lot of different things. So for example, it could be involved in voltage-gated calcium channels. That might be one of the sources. It comes in. It could target, for example, calmodulin. It could target protein kinases to allow for phosphorylation of proteins. It can involve ion channels. There's a lot of different effects that these second messengers can do. Cyclic AMP, again, the source could be from adenylate cyclase, and then taking it from ATP, so it allows for cyclic AMP production. Again, it could also allow for activation of protein kinases. It could also affect cyclic nucleotide gated channels, so it could open up different types of channels. Then you have cyclic GMP, which is going to be very similar to cyclic AMP. Then you have IP3. What does IP3 stand for? It's inositol triphosphate. So we'll just call it IP3. And that's going to act on PIP2. What does PIP2 stand for? Phosphatidylinositol bisphosphate. That's what PIP2 is. And that's going to be activated by diacylglycerol. So don't bog yourself down with all those targets of these different second messengers, but just be familiar with what these second messengers are. So if on the exam, for example, you saw a list of these different types of molecules, know that they can serve as second messengers in neuronal intercellular communication. So what does calcium do? Calcium does a bunch of things, right? Calcium can allow for protein kinases to get activated. It does so by binding, for example, to calmodulin. There are things that also regulate the amount of calcium within a cell. So calbindin, which is basically a calcium chelator, can reduce the amount of calcium inside the cell. You can also see things like IP3 increasing calcium levels within the cell. So whenever you see the brackets right here, brackets around calcium, and you see a little I, that means inside the cell. So that's intercellular calcium levels. The ryanodine receptor is going to open up calcium channels. For those of you who took anatomy and physiology, where have you seen the ryanodine receptor before? Oh, really? It's found in muscle. So whenever you have depolarization of a myocyte, that's going to open up, it's going to be activating the ryanodine receptor, and then that allows for calcium to flood out of the sarcoplasmic reticulum, right? Remember, the sarcoplasmic reticulum is where you have calcium stored up within the muscle, so it can be released intracellular to allow for muscle contraction. So the ryanodine receptor can also be involved in calcium regulation within the cell. Calcium is also, if you remember from how neurotransmitters get released, when you have like the neurotransmitter vesicle binding on to the presynaptic cell, the axon, and it releases neurotransmitters into the synapse. It's going to require synaptogammin, and so this is going to be a calcium-dependent protein, and so it allows for neurotransmitter release. So I didn't include this in. Actually, you guys have the updated slides now, so you have this on there. So synaptogammin, it's hard to see, but it's going to be this little tiny protein over here, and it latches on to the membrane at the end of the axon. So it's going to be part of the presynaptic terminal. And so neurotransmitters can be released in the presence of calcium. Recall also when we're discussing neurotransmitter release that calcium was really important for that process. Without calcium, you're not going to get the release of a lot of your neurotransmitters. So now let's talk a little bit about cyclic nucleotides. So we already addressed some of these. So we have adenylate cyclase. Adenylate cyclase is what allows for the production of cyclic AMP, but you also need ATP for that process to take place. Guanylate cyclase is very similar. It does a very similar thing. It's going to produce cyclic GMP from GTP, so guanosine triphosphate instead of adenosine triphosphate, but basically does very, very similar things. And then what do? They effectively allow for protein kinases to be activated. And what do protein kinases do? They allow for the phosphorylation of transcription factors to allow for gene expression, right? That's how you get transcription of RNA and then ultimately translation of proteins that you're trying to produce. So here are a couple other examples of your second messengers. So you have DAG, which is diacylglycerol, but we're going to refer to it as DAG or DAG from now on. And then you also have your IP3 molecule. So how do these work? So first you have your PIP2. PIP2 is then going to stimulate phospholipase C. Once you stimulate phospholipase C, it's going to stimulate diacylglycerol or DAG, and then that ultimately is going to allow for your protein kinases to be activated. And then similarly to the other processes, you're also going to get gene expression from your protein kinases. IP3 is going to be associated with releasing calcium. And so calcium is also going to be having its own downstream effects. In this case, it would be considered a third messenger instead of a second messenger. But calcium does act as a second messenger as well. as we've already established. Here are some images from your textbook. This is FYI. I'm not going to hold you guys accountable for any of the stuff, but yeah, I'm not even going to go into it really. So let's just skip through this. Not super high yield. But protein kinases versus protein phosphatases, that is high yield. So that's something you're going to see on your exam. So protein kinases, again, protein kinases phosphorylate proteins. They take an inorganic phosphate and they basically attach it to a protein, and that's what allows for the protein to get activated. To reverse that effect, that's where protein phosphatases come into play. They're going to dephosphorylate the protein. And there's going to be different residues that are going to be associated with these. So we're going to talk about like tyrosine kinases. There's also serine and threonine kinases, but different cascades, but similar concept. Right? They're going to be involved in the phosphorylation of proteins. And then you have other residues on certain enzymes as well that allow for phosphatases to come into play. Same thing, serine and threonine, as well as tyrosine protein phosphatase. In terms of protein kinases are very particular about how they get activated. So they're more specific. They have to have like a specific signal to activate them. Whereas with protein phosphatases, to remove the inorganic phosphate, they're not as specific. So they can have a little bit more of how would you say? They're a little bit more promiscuous in terms of what kind of proteins they can act on. Protein kinase is more specific, protein phosphatase is less specific. So they can enact their effect on more proteins. Protein kinases, again, that's going to be transferring an inorganic phosphate. And where does it get that phosphate from? It gets it from ATP, right? So it cleaves off the inorganic phosphate from ATP and it attaches that inorganic phosphate onto a protein. It's using, depending on what kinase you're talking about, it can use serine, threonine, or tyrosine. So receptor tyrosine kinases are very, very common. We even talked about some of those earlier on this semester. I told you didn't need to remember those specific protein or tyrosine kinases, but just understand the foundational concept of what these things do. This is reversible, of course. If you put an inorganic phosphate onto a protein, you can also reverse that by removing it with the phosphatase. So here are a couple different examples of protein kinases. Some of these require different types of, I guess you could call them cofactors, right? Enzymes. They need things present for them to actually function. So this one, protein kinase A, is going to be cyclic AMP dependent. So in order for protein kinase A to work, you have to have cyclic AMP. And here's the cascade of events. You have cyclic AMP. It's going to bind to one of the domains on the protein kinase. You get a change in the shape of the protein kinase. Because remember, whenever you hear ASE at the end of something, it's an enzyme, right? And so enzymes help to speed up reactions. And in order for the enzyme to work, in this case, you have to have binding to cyclic AMP. Once it does its job, you're going to see phosphorylation of the substrate. Sometimes it's going to be found within the nucleus, right? So it's going to allow for gene modification. So gene expression. Here's another example of a protein kinase. This one is going to be calcium and calmodulin dependent. So the way that this is written out is CAMK2. So this is calcium calmodulin dependent protein kinase. It's not going to do anything unless it's in the presence of calcium and calmodulin. Then it gets turned on, it gets activated, and then it's going to allow for cellular modeling, memory, right? So this is going to remain active for a while within the cell. And so this could be involved in, for example, memory formation within the cell. Then you also have protein kinase C. For this one, it has to have the presence of DAG as well as calcium to be activated. This one involves a little bit of a different cascade. So protein kinase C is going to move from within the cytoplasm. So it's kind of floating around the cytoplasm, and then eventually it winds up fusing with the cell membrane. It can also be have its effects on the cytoskeleton as well as the nucleus. So lots of different downstream cascading effects that these protein kinases can have. And then here's your receptor tyrosine kinase, so protein tyrosine kinase. There's a couple different types, and I'm not going to test you on the subtypes of these, so don't worry about these subtypes, but there's one that's going to be a homodimer. There's another one that's a non-receptor tyrosine kinase, and that's going to be associated with the receptor, but it's not part of the receptor. Receptor tyrosine kinase is the actual receptor itself. It needs to get stimulated by a ligand. Whereas the non-receptor tyrosine kinase, there's a tyrosine kinase embedded within the cell, but the receptor itself is not the actual tyrosine kinase. What does this do? Receptor tyrosine kinases are really important for cell differentiation, cell growth. It's also going to be important for survival. So it can be involved in like trophic factors, for example. So receptor tyrosine kinases can be involved in, for example, BDNF. You guys remember brain-derived neurotrophic factor. That one actually binds to tyrosine kinase B, if I remember correctly. And so it allows for the growth and survival of the cell. There's some that allow for cell differentiation, because if you have a cell that's not differentiated and you want to turn it into like a specific type of neuron, tyrosine kinases are going to be involved in that process. So let's watch a little video on protein tyrosine kinases.
Hi guys, today we'll be talking about tyrosine kinase receptors versus non-tyrosine kinase receptors. So this is a drawing of both of them, schematic, and I'll have this picture on the right side as we go through each of the steps. So first, let's talk about tyrosine kinase receptors. And as we talk about each step, you can look at the image, and on the image, it's the picture that's on the left side. So in the first step, the ligand is going to bind to that receptor, and this receptor is a transmembrane receptor. It's on the cell membrane. So it's going to bind, and this binding is going to cause phosphorylation of that receptor, specifically phosphorylation on the tyrosine residue, which is on the receptor, is going to be phosphorylated. Now, this phosphorylation is going to cause downstream effects of activating the MAP signaling pathway. Once the MAP signaling pathway is activated, that's going to cause more downstream effects until ultimately it's going to activate transcription factors within the nucleus. And this is how this tyrosine kinase receptor pathway works. Now, hormones, ligands that actually use this pathway include hormones like insulin and growth factor. Not hormone, growth factor. So make sure you know that. Now, let's talk about that in comparison to non-receptor tyrosine kinase. And that's the picture on the right. So non-receptor tyrosine kinases. The ligand is also going to bind on this receptor, which is also a transmembrane receptor. However, there's going to be a key difference here between the two. So when this ligand actually binds, it's not going to cause phosphorylation of the receptor. There's no tyrosine residues on that receptor, but there are tyrosine residues adjacent to the receptor on the JAK. So it's going to cause phosphorylation of the tyrosine residues on JAK. When JAK is phosphorylated, it's going to cause activation of the JAK-STAT pathway, and that's going to have downstream effects of ultimately activating transcription factors in the nucleus. So the key difference here is the receptor itself is not going to be phosphorylated, unlike the receptor tyrosine kinases, which are phosphorylated. And this all happens intracellularly. Now, hormones, ligands that actually use this pathway will include growth hormone. So in this case, it's not factor, it's growth hormone, prolactin, and EPO.
So that's a good video to describe basically the difference between those two effects.
My next guest is a best-selling author who has written such thrillers as Carrie, The Shining, and The Dead Zone, and his new book, which is entitled The Fire Starter. Ladies and gentlemen, please welcome Stephen King.
Should we watch that? Yeah. It's a little bit more exciting to listen to Stephen King than listen to me explaining tyrosine kinases. I'm not going to test you guys on all the intermediary steps. So I'm not going to hold you accountable for like the JAK-STAT pathway and all that stuff. That's too much. What you do need to know, though, is what I highlighted here in red. Cell differentiation, cell growth, cell survival. That's kind of like the ultimate goal of these pathways. I do not want you guys to remember the whole JAK-STAT pathway, the MAPK pathway, unless you want to. I'm not going to test you on those pathways.
How about
Just
the receptor, non-receptor?
Maybe. Most important thing is going to be the ultimate effect of the activation of the tyrosine kinase. So here's another one. Here's your MAP kinase. So mitogen activated protein kinase. That's what MAPK stands for. This is also going to be involved in, so you get activated by growth factors. You get RAS phosphorylated. You get these kinases downstream that get involved later on in this cascade. And then again, ultimately, gene expression. That's like the ultimate goal of this whole thing, is regulating gene expression. Other things that can affect this is stress, right? So heat shock proteins, osmotic stress, different things can affect the MAPK pathway. And then here is the opposite, right? So we just talked about kinases. Now we're going to talk about phosphatases. And phosphatases, what they do is they remove that inorganic phosphate group. Again, there's going to be less specificity in this, right? They're more what's it called? I was going to say polygamous, but they're more, what's it called when you're promiscuous? They're more promiscuous. They work on multiple different types of proteins. High degree of similarity among the catalytic subunits, right? Whereas the protein kinases are more specific, protein phosphatases are less specific. When you hear the term constitutively active, this means that it is ligand independent. It doesn't require activation by a ligand. So that's why it's constitutively active. And they're not going to be associated with second messengers. They basically just act on their own. So here's a couple of different types of protein phosphatases. There's PP1. This one's pretty prevalent. It's going to be involving serine threonine phosphatase. Remember, we have like serine and threonine kinases as well, right? It's going to be involving several different subunits. I don't care about the subunits that regulate this, but also low substrate specificity. That means it's more promiscuous. It can work on different types of proteins very easily. Here's another protein phosphatase. It's PP2A. This one is one of the most abundant ones in the the central nervous system. There's a bunch of different versions of protein phosphatase. There's also, again, similar to the other protein phosphatases, low specificity. This one in particular has been linked to Alzheimer's disease. There's a lot of things linked to Alzheimer's disease. ApoE4 mutation, for example. There's a bunch of genes that can be associated with Alzheimer's, but this is going to be one of those two. So if it's not working properly, you might see Alzheimer's in these patients. Maybe. Protein phosphatase 2B. This one, also referred to as calcineurin. This one's going to be involving calcium signaling, and this could also be associated with the hippocampus, right? So it could be involved in long-term depression of the hippocampus. Not super high yield. Here's another not so high yield one. So this is activation of the PP2B and how it's regulated by calcium as well as calmodulin. It's part of the regulatory units, right? So it can allow for prevention of phosphatase activity. I'm not going to hold you guys accountable for that. So those are your phosphatases. Again, long story short, phosphatase removes that inorganic phosphate and also not super specific. They can operate on lots of different types of proteins. Now switching gears and talking about dendritic spines. Remember your dendrites, those are going to be receiving a little antenna that are receiving signals from upstream axons. What do dendritic spines do? So these are going to be like little tiny like outpouches and little protrusions that come off of the dendrites. They're going to be resistant. They're going to be resistant to signals. And so they help to moderate the excitatory postsynaptic potential. Remember, this is part of the postsynaptic cell, right? And so they can moderate excitatory signals. They're basically going to function as a bottleneck. So they are a bottleneck for ions, for example, like calcium. There's lots of different shapes to these, so like very diverse shapes of these dendritic spines. They can also be a reservoir for different types of proteins. So dendritic spines, outpouching, they might be able to hold things in there to keep maybe storage for whenever it's needed, so they can serve as a reservoir. Here's some not photographs, but illustrations produced by Cajal. Right, so these are some of his drawings of dendritic spines back in the day. This is in the cortex. So within the pyramidal neurons, you can find a bunch of dendritic spines. I'm not going to test you on which regions of the body you find dendritic spines, but whenever you see dendritic spines, know that it's basically helping to moderate your excitatory postsynaptic potentials. And they can also serve as storage units for proteins. Helps moderate the excitatory postsynaptic potential, because this isn't the postsynaptic cell, right? This is the receiving cell. And so as the receiving cell is getting signals from the presynaptic cells, right, it can help to moderate those signals. So you're not getting too much excitatory transmission, right? So it kind of acts as like a buffer. Think of it as a buffer. It reduces intensity of signaling. So here's an example of a postsynaptic density. Within that, you're going to find all these little spines, outpouchings of the dendrites. So lots of diversity in those structures. Here's another image of things that you might find within a dendritic spine. So this is like super low yield stuff. I'm not going to test you guys on this, but know the function overall of the dendritic spine, what the whole purpose is, reducing excitatory postsynaptic potentials. Now, we discussed what those protein kinases do, and then we kind of alluded to your transcription factors and how they are going to come into play to regulate gene expression, right? Allow for transcription of RNA, allowing for the translation of proteins eventually. So this is the whole purpose of these second messengers. It's going to be allowing ligands to affect intracellular changes that can increase the amount of gene expression. And the gene expression is going to be carried out by transcription factors. And so when you get a transcription factor binding to a certain region of the DNA that you want to transcribe, then eventually you get a whole complex coming into play, allowing for RNA polymerase to attach, allowing for the polymerization and the synthesis of RNA. And then ultimately, after that, you get translation of proteins. So what's the process look like? Your chromosomes are going to be highly condensed, right? They're going to be condensed, and then they have to be unraveled, right? So you got all these histones. Histones you think about it as like a spool, right? You guys do any sewing, right? You have a spool of thread, right? Think of the histone as the spool. And in that spool, you get condensing of your chromosomes. And then you have to have uncondensing of the chromosome, and that's when you have those transcription factors actually coming into play to allow those regions of DNA to actually be transcribed. So, how did these take place? Here are some of the things that regulate gene expression. So you have CREB, C-R-E-B, which is cyclic AMP response element binding protein. That's a mouthful, so we're going to call it CREB. It's going to be bound to CRE, which is the cyclic AMP response element. And it's going to be associated with several different types of signals. So you have your protein kinases, like protein kinase A. It could be involved in MAP kinase, which is also referred to as RAS. It could be involving calmodulin kinase. That's supposed to be there's supposed to be a C right there. Dang it. So instead of just an A, that's supposed to be a C right there. So it's calcium calmodulin kinase. My bad. Let me find that slide and fix that real quick. Cool. And in other words, you have lots of different signals, second messenger signals, that are going to eventually converge to one transcription factor. Right? So all these different pathways can lead to one single thing. That's kind of like the ultimate idea of this. You have multiple different signal pathways that can eventually lead to one type of transcription factor. So here are a couple different examples of what you could see with CREB, right? Again, protein kinase A could come into play here. Geez, that's supposed to be calcium calmodulin kinase. Sorry about that. Put a C in front of that. So C in front of A here. So calcium calmodulin kinase, MAP kinase, right? They're going to allow for RNA polymerase to synthesize RNA. And ultimately, you get the translation of proteins afterwards. So you can also find nuclear receptors. These can also act as transcription factors. Nuclear receptors, for example, could bind to things like hormones. So the example here would be glucocorticoid receptor being bound by cortisol. Right? So cortisol allows for changes within the nucleus. It allows for isolation of the area within the DNA that you want to express. And then you have RNA polymerases coming into those areas to allow for the synthesis of RNA and then proteins as a consequence. It could also be not bound to hormones. So you can have transcription factors that are involved, for example, with like thyroid hormone, and then they could act as maybe a repressor, for example. So they repress certain areas that might be preventing transcription of RNA, and then the promoter now is able to actually start operating properly. And then once the promoter region gets bound to RNA polymerase, then you get the creation of RNA. So a couple different ways that this could happen, but ultimately just kind of know transcription of proteins, or sorry, transcription of RNA and translation of proteins. CFOS is a really interesting gene. So this is considered an intermediate early gene. CFOS expression is going to be involved in cell proliferation as well as cell differentiation. So whenever you have CFOS being activated and being triggered, it's going to allow for proliferation of cells as well as the differentiation of cells. It's a gene, and then it also can turn into a transcription factor, and then you get all sorts of like downstream effects from CFOS activation. You also have growth factors coming into play. So here's your nerve growth factor, NGF, which when it's in a complex with tyrosine kinase A, it can do a bunch of different effects downstream, including, for example, survival of the neurons, growth and differentiation. There's a couple different pathways here involved in this, right? So you got your tyrosine kinase receptor binding to a growth factor, right? And then you have downstream cascade of effects. Lots of different things can happen. Cell survival, neurite outgrowth, right? Cell development, cell differentiation. Lots of different effects from these growth factors. I am not going to hold you guys accountable for all these little intermediary steps, right? Don't worry about all the middle steps here, right? All the different pathways. I'm not going to test you guys on that stuff. Just understand the ultimate goal of these chemical signals. Again, I'm not going to test you on these ones over here. This is like a little bit too much for this class. So if you're interested in this kind of stuff, knock yourself out. You can, delve into it in more detail. But like I said, I'm not holding you accountable for this. Yeah. No, thank you. Yeah, this is not molecular biology. This class is neuroscience. We're not going to go into all that minute, gritty detail. That's too much stuff.